Today is Sunday, 18th of May, 2025 Eastern Time. This science and technology update is brought to you by Preeti Capital, your trusted source for intelligent insights. CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, has revolutionized the field of genetic engineering since its development. This groundbreaking technology allows scientists to edit genes with unprecedented precision, efficiency, and flexibility. Originally discovered in the adaptive immune systems of bacteria and archaea, CRISPR-Cas9, the most commonly used system, has been adapted for use in a wide array of organisms, including humans. It works by utilizing a guide RNA to direct the Cas9 enzyme to a specific location in the genome, where it creates a double-strand break. This break can then be repaired by the cell's natural repair mechanisms, allowing for the insertion, deletion, or alteration of genetic material. The impact of CRISPR is profound, with potential applications spanning medicine, agriculture, and biotechnology. In medicine, CRISPR holds promise for the treatment of genetic disorders such as cystic fibrosis, muscular dystrophy, and sickle cell anemia by correcting the underlying genetic mutations. Recent advancements have also seen its application in cancer research, where it is used to identify and target oncogenes. In agriculture, CRISPR enables the development of crops with improved yield, nutritional value, and resistance to pests and diseases, potentially contributing to food security. Moreover, it has facilitated the creation of organisms capable of producing biofuels and other valuable chemicals, underscoring its versatility across various domains. As research progresses, CRISPR continues to be at the forefront of discussions concerning ethical considerations, regulatory frameworks, and its long-term implications for humanity. The transformative potential of CRISPR technology has become a focal point of genetic research, offering unprecedented capabilities in gene editing. At its core, CRISPR-Cas9, the most well-known system, functions as a molecular scissor that can precisely target and modify specific DNA sequences within living organisms. This system was derived from a natural defense mechanism found in bacteria, where it serves to protect against viral attacks by cutting the DNA of invading pathogens. The precision and efficiency of CRISPR have revolutionized the field of genomics, allowing scientists to edit genes with remarkable accuracy. This capability extends across a vast array of applications, from agricultural improvements to human health advancements. In agriculture, for instance, CRISPR has been used to develop crops with enhanced nutritional profiles, increased resistance to pests and diseases, and better adaptability to climate change. These innovations hold the promise of boosting food security and sustainability. In the medical domain, CRISPR's ability to correct genetic mutations has opened new avenues for treating genetic disorders such as cystic fibrosis, muscular dystrophy, and sickle cell anemia. Clinical trials are underway to explore its potential in curing these and other conditions at their genetic root, which could fundamentally alter the landscape of genetic medicine. Despite its promising applications, CRISPR technology is not without challenges and ethical considerations. Issues such as off-target effects, where unintended parts of the genome are edited, and the long-term impacts of genetic modifications are subjects of ongoing research and debate. Moreover, the ethical implications of gene editing, particularly in human embryos, necessitate careful deliberation to navigate the moral landscape this technology presents. As CRISPR continues to evolve, it remains imperative for scientists, ethicists, and policymakers to collaborate in harnessing its potential responsibly. CRISPR-Cas9, a revolutionary gene editing tool, has transformed the landscape of genetic engineering by offering unprecedented precision and versatility. This technology, derived from a natural defense mechanism in bacteria, allows scientists to make targeted modifications to the DNA of living organisms. The core components of CRISPR-Cas9 include a guide RNA, gRNA, and the Cas9 protein. The gRNA is designed to match a specific DNA sequence within the genome, directing Cas9 to the precise location for editing. Once there, Cas9 acts as molecular scissors, cutting the DNA at the targeted site. This cut can either disrupt the target gene or allow for the insertion of new genetic material. The applications of CRISPR are vast and growing. In agriculture, scientists are employing this technology to develop crops that are more resistant to pests and diseases, improve nutritional content, and withstand harsh environmental conditions. In the field of medicine, CRISPR holds promise for treating genetic disorders such as sickle cell anemia and cystic fibrosis by correcting mutations at their source. Additionally, it is being explored as a potential tool for combating infectious diseases by disabling viral DNA within host cells. Despite its potential, CRISPR technology raises ethical and safety concerns. The possibility of unintended off-target effects, where the Cas9 enzyme cuts DNA at unintended sites, remains a significant challenge. Furthermore, the long-term implications of gene editing in humans are not yet fully understood, necessitating rigorous ethical considerations and regulatory oversight. As research progresses, the scientific community continues to refine CRISPR techniques to enhance their accuracy and reliability, ensuring that the benefits of this groundbreaking technology are realized responsibly. CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, has revolutionized genetic research by providing a precise and efficient tool for editing genomes. At the heart of CRISPR technology is the Cas9 protein, which acts as molecular scissors capable of cutting DNA at specific locations. This allows researchers to add, remove, or alter genetic material with unprecedented accuracy. The implications for medicine are vast, with potential applications ranging from correcting genetic defects to treating and preventing the spread of diseases. Recent advancements in CRISPR technology have focused on improving its specificity and reducing off-target effects, which occur when the Cas9 protein cuts DNA at unintended sites. Scientists have developed high-fidelity versions of Cas9 and engineered guide RNAs to enhance targeting precision. These improvements are crucial for clinical applications, where unintended genetic alterations could lead to harmful consequences. In addition to its applications in human health, CRISPR is being used to address challenges in agriculture. Researchers are employing CRISPR to develop crops with enhanced nutritional profiles, increased resistance to pests and diseases, and improved tolerance to environmental stresses such as drought and salinity. This could lead to more sustainable agricultural practices and help meet the food demands of a growing global population. CRISPR's potential extends to environmental conservation as well. Scientists are exploring the use of gene editing to control invasive species and protect endangered ones. By altering specific genes, it may be possible to reduce the reproductive success of invasive pests or bolster the resilience of vulnerable species. As CRISPR technology continues to evolve, it is accompanied by ethical and regulatory considerations. The potential for germline editing, which could lead to heritable genetic changes, raises significant ethical questions that must be addressed through comprehensive public discourse and policy development. CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, has dramatically transformed the landscape of genetic engineering since its mainstream introduction in the early 2010s. This revolutionary gene editing technology, harnessed from a natural defense mechanism found in bacteria, allows scientists to make precise modifications to the DNA of living organisms. Its potential applications span across various domains, including medicine, agriculture, and fundamental biological research. In recent years, CRISPR has been at the forefront of groundbreaking developments in the field of medicine. It has enabled researchers to explore potential therapies for genetic disorders such as sickle cell anemia, cystic fibrosis, and certain types of muscular dystrophy. By directly targeting and repairing defective genes, CRISPR holds the promise of not just treating, but potentially curing these conditions. In agriculture, CRISPR is being employed to enhance crop resilience and yield. Scientists are developing crops that can better withstand environmental stresses such as drought and pests, which is crucial in the face of climate change and the growing global population. Moreover, CRISPR is being used to improve the nutritional content of food, paving the way for more sustainable and healthful agricultural practices. Despite its immense potential, the use of CRISPR technology raises ethical and safety concerns. The possibility of off-target effects, where unintended parts of the genome are edited, remains a technical challenge. Furthermore, the prospect of gene editing in human embryos has sparked significant debate regarding the moral implications of such interventions. As CRISPR technology continues to evolve, it is imperative that the scientific community, policymakers, and the public engage in ongoing dialogue to navigate the ethical landscape and harness its benefits responsibly. The future of CRISPR is undoubtedly promising, yet it demands careful consideration and regulation to ensure its safe and equitable application across various fields.